Good morning, everyone, and welcome to Microbial Minutes, the new and artificial life edition. This is Julie Wolf, the science communications specialist at the American Society for Microbiology's every couple weekly update on what's hot in the microbial sciences. Today is November 27th, 2018. And today we will be talking about a number of things that have been going on in the microbial sciences, including a link between the microbiome and diabetes. We will talk about the State of Malaria Report released by the World Health Organization. Then we're going to move into the part about the new and artificial life. First, we'll discuss a report on a potential new kingdom of eukaryotic life. Very exciting. And then artificial cells, or cell mimics, as the authors call them. Let's dive right in. We're going to start with two reports, one from Science Translational Medicine and one from Cell, about the microbiome's role in diabetes. And the take-home message from both of these stories is that microbially derived products influence diabetes and the pathogenesis of diabetes. Diabetes is a very severe problem. It's a worldwide phenomenon, and within the US, there are over 30 million people who have diabetes. Up to a quarter of those people are unaware that they have this condition. However, they may suffer from some of the effects, such as feeling a little sluggish after meals, but they haven't been properly diagnosed. And that doesn't even consider the over 84 million people who have prediabetes that are in a dangerous stage where they may yet develop full-blown diabetes. Now, most of the people in the United States and elsewhere have type 2. 95% of the United States cases are type 2 diabetes, with the remainder being type 1. And the difference is illustrated here on the right-hand side. Type 1 diabetes is a genetic condition where one does not make insulin. So the islet cells of the pancreas don't make insulin. Insulin is a signal to cells that there is food around and they should take up that food. And the food is glucose. So as we eat food, we digest it, we turn it into its basic components, one of which is glucose. Our blood sugar rises, and that sugar needs to be taken up by cells. The cells are signaled by insulin to take up that sugar. If you don't have insulin, there's no signal to take up the sugar, and that's type 1 diabetes. Type 2 diabetes, patients do make insulin, but it's either not a sufficient concentration to signal to cells, or the cells don't respond properly. Perhaps that insulin isn't processed to a form that the cells recognize, or perhaps the cells don't respond to the insulin. So there is both sugar and insulin in the bloodstream of type 2 diabetic patients. Now, the number of Americans diagnosed with diabetes has tripled over the last 20 years. So this is a growing problem. And these scientists that wrote both of these reports wanted to ask about the role of the microbiome in type 2 diabetes. So in this more prevalent form of diabetes, illustrated here on the right. They're going to take a couple of previously established facts into consideration. One is that insulin resistance increases with age. So the older you are, the more likely you are to have insulin resistance and not respond to that glucose taking up signal. And the second fact is that the microbiome structure changes with age. So there's different proportions of the bacteria that are part of the microbiome, the gut microbiome. As you get older, the proportions or the ratios of those bacteria change. And in particular, we're going to focus on the commensal bacterium Akkermansia muciniphila, which varies with age. This is a gram-negative bacteria. It's slightly immunostimulatory. It activates TLR2 responses to have a little bit of innate immunity, but it also metabolizes dietary fibers into short-chain fatty acids, things such as butyrate, acetate, propionate. These are necessary for gut health. We're going to take another previously established fact, and that is that insulin resistance is induced by activated innate 4-1BBL positive B1A cells. This is a subset of the B cells, which are part of your white blood cell immune system. And this subset is associated with insulin resistance. So the loss of Akkermansia muciniphila impairs intestinal integrity. And this is where the scientists have some very beautiful experiments demonstrating that when this bacterium is not around, the tight junctions between the epithelial cells become less tight, and bacterial products, things like endotoxin, can leak outside of the gut. And endotoxin is very inflammatory and immunostimulatory, but in the absence of some of these short chain fatty acids, in particular butyrate, the immune system is not quenched. So butyrate, when it is present with some of these bacterial products, if the gut becomes a little leaky, will help to quiesce or dampen that immune response. But because the in mice that do not have this Akkermansia muciniphila, there is no butyrate being produced or less butyrate being produced. CCR2 positive monocytes are activated. CCR2 positive monocytes are a subset of the innate immune system. And what these activated monocytes will do is to convert some of those B1A cells into that 4BL cell, or the insulin resistant associated B cell subset. So I have a couple of the major experiments that they used to demonstrate these facts. And here we have young mice and aged mice, and they looked at the amounts of Akkermansia muciniphila by investigating their microbiome, and they saw that the aged mice have less of this Akkermansia muciniphila, and they also have a higher blood glucose concentration. So the young mice have a something around 300 arbitrary units, whereas the aged have over 350. But this concentration of blood glucose can be brought down by application of antibiotics that are specifically administered to promote the growth of Akkermansia muciniphila. So these antibiotics would allow the ACK, or Akkermansia muciniphila, to grow to higher levels, to generate butyrate, and to quiesce that immune system. And this will help to bring down the blood glucose levels. They also showed that the insulin resistance is reversible by addition of the bacterium itself, by addition of butyrate, so the microbial derived product, or they could take an antibody to deplete the monocytes that become activated. So here we have the young and the aged mice. Here, instead of looking at blood glucose, we're looking at insulin, plasma insulin concentrations. And you can see that the aged mice are almost twice as much, have almost twice as much plasma insulin. If the researchers added a nonspecific antibody, this is not supposed to have any effect. There's still higher levels of that plasma insulin, but if they add antibodies that will deplete that monocyte population, that CCR2 positive monocyte population, they can bring down the plasma insulin levels and rescue the insulin resistance phenotype. So the second story is from Cell, and this one is going to take into account the fact that people who have type 2 diabetes have higher imidazole propionate levels. So propionate, as I just mentioned, is one of the short chain fatty acids that is generated from some of our dietary fibers, but it's also a product of histidine biosynthesis, and this is not propionate itself. This is a modified version, imidazole propionate, so it has a little imidazole group here, and this molecule is associated with impaired glucose metabolism. And the researchers here are going to show that the fecal microbiota are where this imidazole propionate comes from. So they took the fecal microbiota from people who have type 2 diabetes, and from, of course, people who do not have type 2 diabetes, as shown here in the graph on the right. And they saw that the components, the bacteria that are associated with the fecal microbiota of people who have type 2 diabetes, convert histidine into imidazole propionate at a higher concentration. So here we have two individuals making with the addition of histidine, turning that histidine into the byproduct imidazole propionate associated with glucose non-responsiveness, compared to those that do not have diabetes down here. Imidazole propionate, when administered itself, impairs glucose tolerance in mice. And it does this through activation of the mTOR pathway. So it impairs the insulin signaling at the level of insulin receptor substrate, activates the P38 gamma MAP kinase, and eventually promotes P62 phosphorylation and activation of mTOR. mTOR is like a master regulator of cell metabolism. It tells the cell whether it should be actively replicating, whether it should be entering a stage of something like starvation and activating certain processes such as autophagy. And so this becomes quite cyclical, where the mTOR is inaccurately activated, and then the cells do not properly respond to glucose in the bloodstream. So these were not widely shared in mainstream media, but the stories were picked up by some science-specific outlets. Science News wrote up about the gut bacteria, which may guard against diabetes that comes with aging. They had a lovely photograph or electron micrograph of the Akkermansia mucinophila, as you can see here. And it was shared by a number of people on social media as well. On Twitter, Eric Topol communicates all types of things about biotech and biomedicine. Christina Campbell, who's a specific reporter about microbiome science, she just published a book about microbiome specific diets. And Aaron Siegel, who we've actually covered some of Siegel's work on previous Microbial Minutes, and he does quite a bit of work with the gut microbiota, so they were sharing it among their network. All right, moving on, we're going to talk about the World Health Organization and its recent World Malaria Report. Every year, the WHO releases a report about the state of malaria, and my take on this is not a positive one. Sadly, it's that bad news regarding the fight against malaria, but we'll discuss whether it truly is bad news at the end of this story. So malaria is a huge worldwide problem. Nearly half the world's population is at risk of contracting malaria. In 2015, the year I could find the most recent numbers for, there are 212 million malaria cases and 429,000 deaths due to malaria infection. Now, this is a 200 plus page report, so there's a lot of information in here. But one of the take-home messages is that the world is not on track to achieve two of its critical 2020 milestones, which were set by members of the World Health Organization. And one was to reduce malarial deaths by 40% by 2020, and the other was to reduce disease incidence, or the number of people who are contracting the disease, by 40%. This is illustrated by the fact that of all the countries that have heavy malarial burdens, only India has shown progress in decreasing the disease incidence. In India, the cases have dropped 24% in 2017 compared to 2016. So that is a sliver of hope that with the pressure and funding, that incidence and disease can be decreased. So as I mentioned, this is quite a thorough report. It covers all types of things, including the burden, how to prevent it, and how effective those preventative measures are, different ways of diagnostics, surveillance systems that are necessary, as well as different drug treatments and regimens that are necessary to treat and eliminate malaria. And finally, it ended with challenges in getting the malaria response on track. And I've just pulled out a few of the facts. There are both FAQs if you want to just get the surface level, or if you want to get a several page report summary. There were a few different ways that this report was written up that you can find directly on the WHO website. One of the things that I think is a more negative point, or a disappointment, is that the 10 highest burden countries in Africa reported increases in cases of malaria in 2017 compared with 2016. Another quote, in 2017, there were an estimated 435,000 deaths from malaria globally, compared with 451 estimated deaths in 2016 and 607,000 in 2010. So it's definitely decreased over time, but the rate of decrease has not been as large as it has been in the past. And that's in part because of those challenges in getting malaria on track through funding. We've discussed in previous sessions how neglected tropical diseases need a huge influx of funding from NGOs and from developed countries in order to maintain the fight against these diseases. And here we see that in 24 out of 41 high burden countries, which rely mainly on external funding for malaria programs with an E because it's written with British English, the average level of funding available per person at risk declined in 2015 through 2017 when compared to 2012 through 2014. This ranged from a 95% reduction in the Congo to a 1% decrease in Uganda. So that illustrates that maybe it was a hot to fund malaria a few years ago, but those funds have either dried up or waned over time, and unfortunately, the people who are paying the price are the people who are most susceptible to malaria. And this last quote illustrates some of the difficulties in treating malaria. So, with increases in diagnostic testing is cheap and relatively easy to do nowadays for malaria in undeveloped or low resource areas. Artemisinin based combination therapy, or ACT, treatment courses are becoming more targeted toward patients who tested positive for malaria. This is demonstrated by a substantially reduced ratio of ACTs to tests, which is good. We want people who test positive for malaria to get this artemisinin based combination therapy. And artemisinin is one of the most effective compounds against malaria, but resistance has been cropping up, and so it's now being used in conjunction with either one or even two additional compounds to treat people who have malaria, just to make sure they aren't able to develop resistance. Nevertheless, to finish the quote, this implies that an estimated 30% of patients who receive ACTs are not tested for malaria, and this bodes poorly for a couple of reasons. Number one, these patients may not have malaria; they may need to be treated for another disease at the onset of disease. A lot of diseases or illnesses will present with similar symptoms, and it's important that we treat diseases as they are needed to be treated. For example, bacterial diseases would need to have an antibiotic administered. And anytime that an anti-malarial or any type of antimicrobial drug is given inappropriately, it increases the amount that's excreted from the patient in urine and feces and is put into other systems, and increases the potential for low-level exposure of the microbe and development of resistance. And resistance is a big problem with malaria in certain areas of the world, particularly in Southeast Asia. If you go to the furthest south area, you can see that high ACT failure, which is over 10%, to up to 4 different compounds at once, seen in Cambodia, 3 different compounds in Thailand. And so this is definitely something to be concerned about. However, a new report, now I want to transition to a report from the New England Journal of Medicine, shows that artemisinin-resistant Plasmodium falciparum is not only found in Southeast Asia. Eastern India has now developed a new type of resistance, or a previously non-described form of resistance. And so in this case, the doctors wanted to ask, or the scientists wanted to ask, what proportions of their patients are clearing malaria when they're treated, and how long does it take for patients to clear those malaria parasites from the blood. They could actually follow the patients as they're administered one of these artemisinin combination therapies. They give them to 136 different patients, and then every 6 hours they would make a thick blood smear to see what type of parasites were still around in their peripheral blood. And they saw that most patients were able to clear the parasites within 2 to 4 hours, which is pretty good. But that a small proportion took between 5 and 7 hours. And so they then looked at the Kelch13 genotype. And Kelch13 is a gene in Plasmodium falciparum that is associated with different types of resistance. But these particular mutations had not previously been associated with resistance. And so here we're looking at whatever the more common amino acid is, an N here, and the place in the gene, the 29th amino acid residue, is now an L. Similarly, an F at 446 is now an I, etc. And what they saw is that when they separated based on the Kelch13 genotype, there were this R539 and this G625 these two mutations, which had not previously been described, were associated with a parasite clearance half-life of over 5 hours. So they were most likely representing this 5 to 7 hour group. And that's important to note because beyond the 5 hour half-life, efficacy of this combination therapy drops, and this is more likely to be associated with artemisinin resistant malarial parasites. And so, as the authors themselves say in their letter to the New England Journal of Medicine, our findings emphasize the need for increased surveillance of drug resistance in order to manage the spread of resistant parasites, because not only is resistance spreading, but it is spreading through mutations that have not previously been reported. So these studies, particularly the World Health Organization report, were written up in a number of different outlets. The New York Times used the headline that the fight against malaria has reached a standstill, perhaps a more neutral take than my bad news. And they spoke with a head of a malarial research unit who has said that this triple therapy should become the standard for all treatment as a way to combat resistance. So using three drug combinations seems to be more effective than using two. And then the Telegraph used the headline countries hardest hit by malaria see rise in cases. So this seems to be, unfortunately, on the negative side, negative take from this World Health Organization. And this this article really focused on the challenges in funding with the CEO of a nonprofit called Malaria No More UK, emphasizing that the countries where malaria takes place need to take charge of some of the especially low cost preventative measures. Things like bed nets are relatively low cost to implement, but the distribution can be quite difficult. And this particular CEO says that the countries and the governments within those countries should take charge of making sure that those types of preventative measures are put in place. Moving on, we're going to enter into the new and artificial life portion of our session. We're going to start with this exciting report from Nature, which is titled Hemimastigophora is a novel supra kingdom level lineage of eukaryotes. Take home message is that this microbe that has been discovered may be a new kingdom level category of life. Humans love to put things into categories. We categorize all types of life forms into various groups and subgroups. You may remember from elementary school, dear King Philip came over for good spaghetti or from good Spain. There's a number of different anagrams, and each of those letters stands for one of the categories into which there are even more categories. So the deer is the D, which is a domain, which represents the three domains into which all life is categorized. We talk a lot about archaea and bacteria on this in these Microbial Minutes sessions, but of course there's also the eukarya, which represents both us, human beings, as well as single-celled eukarya, such as the fungi and the protists. Now, below domain is kingdom, and there are, depending on what categorization system you're using, 5 to 8 categories. Here I'm talking about the 5 different categories: animals, plants, fungi, algae, and protists. Protists have typically been used as a catch-all category for things that don't really fit into one of those other 4 groups: animals, plants, fungi, or algae. Now, this group from Canada suggests that the hemimastigotes represent a new kingdom level category. So this would be animals, plants, fungi, algae, protists, and hemimastigotes, which is quite exciting to think about an additional category in the kingdoms of life. So this Hemimastigophora, this is not a completely unknown type of organism. It was previously a phylum level category. But what this group was able to do, instead of just that we're in the sequencing era, they were able to do some phylogenomic analyses. They found some of these organisms, and they were able to do single cell analyses. And they found that it was so different, that their sequences were so different that they should potentially be placed outside all other established eukaryotic supergroups. They pointed out that using single cell transcriptomics allows better coverage of housekeeping genes, which is, as you can see over here in the chart on the right, how they were able to separate this hemimastigophora from other types of related single-celled organisms. That's because the housekeeping genes, things such as myosin and metabolic genes that are found in common in many different life forms, are used as markers since they are conserved, but the sequence that is used to code for them may be slightly different. And in fact, they liked this even better than single cell genomics because of the high coverage of those housekeeping genes. They emphasize that these single cell techniques are really ideal for things such as the heterotrophic protozoa, which these organisms fall into, that likely represent major undiscovered lineages. So these images on the right-hand side, these are actually two different species. These are the two species which are indicated here in red, the Hemimastigophora. And they may not look extremely different by eye, and it's important that we use sequencing technologies to determine whether they are the same species or even the same genus, versus being different. Doing things by sequencing allows a finer detail of differentiation than simply looking and doing things phenotypically. This group was also the first to be able to grow hemimastigote culture. They were able to grow this one on the right hand side, Hemimastix kujawezjike. And kujawezjike, it derives from the Mi'kmaq First Nation of Nova Scotia, which is the indigenous peoples of this region of Nova Scotia where the research was done. Translates to little ogres. So this guy is named after little ogres. And this was quite an exciting finding. It was written up in a number of mostly scientific outlets. So Popular Mechanics wrote up scientists discover a whole new kingdom of life, highlighting, importantly, that while the scientific group suggests that this is a new super kingdom level of life, you cannot just simply use one report to add an entire category. This decision can't be made lightly, still has to be verified by other scientists, but it does highlight just how incredibly diverse life can be. So shared in several different places on social media, both by aggregate groups and scientists who summarize the research as go on a hike, end up studying something that forms its own kingdom in the tree of life. And this story, this one by the CBC, which is Canadian Broadcasting category, was really great. Of all the things that I read for today's session, this is the one that I enjoyed the most. It really tells the story of these trainees, graduate students and postdocs and research technicians who went camping, picked up some dirt, rehydrated it, and recovered these new types of life forms. And it also tells the story, I think, in a nice way. It accurately summarizes the importance of a new kingdom level lineage, and it tells the story of how they're able to use microscopy to observe what these hemimastigotes were eating. So they saw, here is one of those hemimastigotes in this microscopy image. It's eating a diatom, which is another small microscopic organism. And once they figured that out, they were able to, as the article says, feed and breed the captive organism, able to domesticate it in a way. And that will, of course, be useful for future studies. And if you want a short 8-minute interview with the senior scientist, you can click on that CBC link that we have below and listen to a short 8-minute interview about this new branch on the tree of life. All right, we're going to move into our last story, and this is something from BioArchive. And often I wait until in BioArchive, which is where preprints for biology can be published. I wait until these have been published in their final journal, final scientific journal, undergone peer review, etc. But this article I thought deserved to be highlighted now for a number of reasons that I'll get to at the end of this story. The title of this is Communication and Quorum Sensing in Non-Living Mimics of Eukaryotic Cells. The take-home message is that new artificial cells are the closest yet to true eukaryotic cells. So I believe we have talked in previous sessions about a minimal genome, things such as SYN 1.0, SYN 3.0. These are the synthetic genomes that were chemically made by Craig Ventnor's group. And testing those genomes, seeing whether those chemically synthesized DNA molecules will function correctly, has always required pre-made cell parts. You have to have basically a bacterial cell in this case that has had its own DNA taken out, but it maintains its cell membrane. It keeps the ribosomes and the other cell machinery that are important for testing that DNA. And so, on the other side of this synthetic biology story, scientists have been working for decades, really, on how to design an artificial cell, starting with, of course, a membrane. One of the most important things that a cell mimic, as the authors of this story call it, cell mimics have to be able to differentiate the inside and the outside. They have to be able to regulate the inside and the outside. They have to allow certain things to come in and certain things to go out. And it can't just be by simple size exclusion, because it needs to be able to differentially choose what goes in and what goes out. They have to be able to store information. In the case of eukaryotic cells, that needs to be in some sort of nucleus-like organelle or organized sub group within that cell. And to really be a true cell mimic, to act like a cell, these cell mimics must be able to communicate with neighboring cells. Imagine something like quorum sensing. As the cell population increases, the cells produce a certain compound, which then allows the activation of genetic programs and the changing of gene expression. And so a true cell mimic would be able to have this type of system built into it as well. So this group describes using microfluidic production of these double emulsion droplets to make their cell mimics. And this is very interesting. Here's a schematic of how they are able to make these cell mimics. Here, the inner aqueous layer, there's that outer aqueous layer, and then this middle organic layer. So they make this double emulsion. And here, they have to polymerize it using UV, and then they dehydrate it a little bit using ethanol and add a HEPS buffer. And during this stage, they have the formation of a nucleus, which is not made of membrane. It's so interesting. It's not made of lipid. It's a clay DNA hydrogel that mimics the nucleus and the DNA content. It contains the DNA, which has been um, uh, put into the cell mimic in this middle organic layer here. And so they do all kinds of really neat assays. If you want to see some of the ways that the cells are able to activate certain gene responses, go ahead and click on the link to the BioArchive piece. One of the really cool things that they're able to do is to show that the cells can respond to density. So here they have a reporter that only responds to high density cell concentration. It's going to have an output of making a fluorescent molecule, GFP, green fluorescent protein, only when the cells reach a threshold density. So here we see what the cells look like in their droplet. Here we have a low density, only 94 cell mimics. Here we have high density, 404 cell mimics within this droplet. And you can see visually that the GFP is only activated when these cells are at that high concentration. And if they look at the cell mimics per droplet, you can see that the intensity of that fluorescence increases as the number of cell mimics per droplet increases. So much, much higher over here. They have another system where they have the same type of GFP response, so that the green fluorescence will be activated in high concentration, but they also have an M cherry that is on all the time. So this is constitutive expression of M cherry, and that means that we should always be able to see the nucleus-like area. I'm putting nucleus-like in quotation marks, where here we have the purple or M cherry positive signal at all times, and then only as the cell density increases do we see the green fluorescence turn on as well. And so they show this, the green to red ratio changes as the red is the only thing that is expressed. I hesitate to even use the word expressed. Is expressed the way that I imagine expressed is the correct term, but that only the M cherry is expressed at a low cell density, and the green is gradually turned on at a high cell density. Very exciting. And so this was, I wanted to share this now before it is published in its final place, because this paper has already been written up in some um, very nice reviews in both Science and Nature, other science outlets, about the ability of biologists to create these artificial cells. So Science wrote this up. Biologists create most lifelike artificial cells yet, in which they say that the latest pseudocells look a little bit like natural cells, but they are made of completely artificial materials, as the co-author uh, Henrika Nieder-Holtmeier is quoted. And Nature has this really lovely piece about how biologists are creating lifelike cells from scratch by Kendall Powell, who, oh goodness, this author, I forget which author this is that they're quoting, but the author says, I've always been fascinated by this question, what distinguishes life from non-living matter? And that's something that I think fascinates a lot of synthetic biologists, who often use microbes as the type of living matter that they want to mimic. And the Nature piece also has some very lovely schematics. So I recommend reading that if you're at all interested um, in this subject and want to learn more. This was shared on social media, despite being BioArchive and not yet published in an official publisher, quote unquote, just yet. Kate Adamala is a synthetic biologist at University of Minnesota who says that this is the closest anyone ever came to engineering an artificial eukaryotic cell. So this is very exciting news for synthetic biology, and certainly something that we'll follow up with in the next in future microbial minutes. All right, I'd like to summarize. Oh, I'm sorry. And if you're more interested in the minimal genome side of synthetic biology, the ability of scientists to chemically design and say exactly what the genes are that are necessary for life, we will also link a blog, Does a Minimal Genome Exist?, written by one of our microbial science bloggers, Jennifer Tsang, and we will link that, of course, as well. All right, to summarize what we've learned today, in the last couple of weeks, we've had microbially derived products found to influence diabetes and the pathogenicity of diabetes. We have found out some bad news regarding the fight against malaria in terms of both funding and the malarial incidence, number of cases, new cases. We found that we learned that one microbe, the hemimastigotes, may actually be a new kingdom level category of life. And then we finally heard about some new artificial cells that are the closest yet to eukaryotic cells. I'd like to thank everyone for listening, and invite you to leave a comment or question if you liked if you liked this session and you want to make sure not to miss any. Go ahead and subscribe so that you're always notified when we have another Microbial Minutes.